Okay. I hope you're seeing the slides and not any other random things, but yeah.
We are seeing the funny business in which you're involved in. Hey, man, seek help, Sarah.
I'll do that. I'll seek help from you, Daktari. So, guys,
Uh-huh.
on Monday, you took us through glycolysis.
Mm-hmm.
Just give us a 1 minute recap on the glycolysis part.
1 minute recap. So
yes, 1 minute recap.
So glycolysis, as we said, it's a breakdown of glucose, glucose lysis, breaking down. So why are we breaking down glucose? To get energy. So glycolysis is whereby glucose is our starting, you can say, ingredient, and we, through a series of enzymatic reactions, we obtain pyruvate if it's oxidative, or if it's aerobic metabolism, that means oxygen is there in good amounts. And if it's anaerobic metabolism, we will go and obtain lactate. Then we say that then we use 2 ATP molecules in glycolysis, and we make 4 ATP molecules in glycolysis. So that tells us we make a net of 2 ATP molecules. Then the last thing we mentioned was on the rate limiting step, which was the third step, the conversion of fructose 6-phosphate to fructose 1,6-biphosphate, whereby we say that is the rate limiting step, which is catalyzed by phosphofructokinase. So if you want to stimulate glycolysis, one of the trigger was AMP. The other trigger was fructose 2,6-biphosphate. If you want to inhibit glycolysis, it was citrate and ATP. I think that's the summary I can give.
Okay, so yeah, so basically it's almost so today we are doing gluconeogenesis, which is almost the opposite of what Guy had said. Actually, it's the same process, but there are a few differences, a few enzymes here and there. Yeah, then, but it's just about the same. So basically what happens generally, there's, since, like, let's say during the day you keep eating here and there, then you have glucose to sustain you. But like, let's say during at night, you're not eating, there are so many hours, so your body generally goes low on glucose. But then other organs like the brain need glucose to function properly. So then we will need to come up with a mechanism to ensure you have glucose throughout the night until you take your next meal. So that's where gluconeogenesis comes in. So basically, we are just trying to get more glucose into the system. So in gluconeogenesis, it's basically just converting pyruvate back to glucose. So that's what we'll be looking at now. So now pyruvate, which is what is going to start our process here, can come from a few places. Like there are a few molecules like lactate, which are glucogenic. So they'll be converted to pyruvate, which will be converted to glucose. So other molecules that can be converted to pyruvate would be something like some amino acids like alanine, also glycerol. Yeah, so they can be converted to pyruvate, which can then be converted to glucose 6-phosphate. But then now, since like for when we were doing glycolysis, we saw like the last step in glycolysis, it's not a reversible reaction. So we just can't reverse the process and start moving back. We need to come up again with a mechanism that works well for us there. So basically what happens in gluconeogenesis, pyruvate is, so glycolysis takes part in the cytosol, yeah? So the pyruvate will be transported into the mitochondria. Then it will be converted to, you remember when we were doing, what else, what other cycle did we do on Monday? TCA, we also did TCA. So the way pyruvate was being transported, then converted, yeah, so it's the same thing. So the pyruvate will be transported into the mitochondria, converted to oxaloacetate, then oxaloacetate transported back into the cytosol, which is now what will continue with the whole process. Yeah, so basically that's one, yeah. So then the enzyme we are using there is pyruvate decarboxylase, which is not found in glycolysis. So in this, what is it called? Whatever. In this, they call it a flow chart. Oh, yeah. So the enzymes that are in, is it red? The ones that are in red, they are only found in gluconeogenesis. Then the other one, they are both in gluconeogenesis and glycolysis. Yeah, so there's that step, the one for pyruvate carboxylase, and the one for phosphoenol pyruvate, fructose 1,6-bisphosphate, and the other one there. Those one are not found in glycolysis, they are just here in gluconeogenesis. And then also, like we said, like Gaius has just told us when he was doing the recap about the rate limiting step in glycolysis, and he's told us about how like ADP and AMP would activate. So basically they would drive for glycolysis, but then when we have more of ATP or citrate, then they would inhibit that reaction. But in them inhibiting glycolysis, they are basically just helping us push for gluconeogenesis. What else do I need to mention for this one? Yeah. Oh yeah. So pyruvate decarboxylase requires a cofactor, biotin. And on Monday, Gaius was asking what's the difference between a cofactor and a coenzyme. So basically, cofactors are, coenzymes is part of a cofactors, like it's a subclass of cofactors. Coenzymes are organic. Then cofactors, they are now the whole umbrella of those things that binds to enzymes and activates them. Yeah, so now for the regulation, it happens at still the same step, but now it's now the enzyme there is now fructose 1,6-bisphosphatase instead of phosphofructokinase like in glycolysis. Yeah, so this now just, at least this gives a good summary of what the 3 steps that are different from glycolysis and what factors would stimulate or inhibit each of them. So we'll look at, on Monday also, Hiral was asking about the Cori cycle. When we are doing specialized tissues, when you're doing muscle, we'll mention something about it. Yeah, like we're saying, lactate, alanine, glycerol, and a few other molecules, they are glucogenic. So they'll be converted to pyruvate, then the pyruvate will be converted to glucose. So that's it. Yeah, that's it about gluconeogenesis. Then now the pentose phosphate pathway, it's also very much related to glycolysis. It's just like a small shunt within the glycolysis pathway. Let me see if there's a yeah. And like the name says, it's a pentose phosphate pathway. So basically pentose 5. So basically it's going to give us the 5, what are they called? 5 carbon molecules. So 5 carbon sugar molecules. And it also gives NADPH in the process. And it happens in the cytosol. Okay. Yeah. So this is part of the glycolysis pathway, and it's called a starting, whatever, the starting molecule for pentose phosphate pathway is the glucose 6 phosphate from what is it called? From glycolysis. So the pentose 6 phosphate, now depending on the body needs, it can either be converted maybe to, whether it's the 5 carbon molecules, if we need, maybe it's for those rapidly growing cells, you need to be replicating and such kind of things. So then you'd produce more of the carbon, those 5 carbon sugars. If let's say the body is experiencing maybe a situation where there's a lot of reactive oxygen species, then you'd need to go towards producing more of the NADPH. If maybe you need more energy, then you'll just go back to producing glyceraldehyde 3 phosphate and glucose 6 phosphate. No, not glucose 6 phosphate. Glyceraldehyde 3 phosphate and fructose 6 phosphate, which again will continue in the glycolysis pathway to give us more of the ATP to be used for energy. Yeah. So pentose phosphate pathway yields NADPH, and basically the 5 carbon sugars, we are talking about ribose 5 phosphate. So those are the basic or the most important products of the pentose 6 pathway. Pentose phosphate pathway. Yeah, so then what else? So pentose phosphate, it happens in the cytosol. What else? Yeah, it happens in two main phases, an oxidative phase and an non-oxidative phase. The first phase is the oxidative one. Its reactions are mainly, what is it called? Non-reversible. Yeah, its reactions are mainly non-reversible. Then the second phase, the non-oxidative phase, which will now give us the fructose 6 phosphate or the glyceraldehyde. Yeah, that one, the ones that goes back to the glycolysis for giving more energy. They are generated in the non-oxidative phase. If you need the sugars for DNA and those replicating cells, then the first phase is more used. Yeah. So yeah. Um, so now that we are here mentioning NADPH every other step of the way, NADPH, it's produced in this rapidly, whatever. Yeah, in this rate limiting or the regulatory step. And then now this NADPH is very crucial for, like we said, what is it called? Dealing with those reactive oxygen species thing is. And you remember, I don't know, about the, what was it? Let me just go back. Glucose 6-phosphate deficiency. When you have that, then what pathways would be affected, guys?
Pardon?
What does glucose 6-phosphate dehydrogenase do?
It's involved in the first step of gluconeogenesis to convert glucose 6-phosphate from the one from glycolysis to phosphoglucone or lactone. So that's, so if you have a deficiency of this enzyme, then you'll be at risk of lots of things, such as these reactive oxidative species, as you've mentioned, lysis, red blood cells, all these things.
Yeah, so basically that reaction will give rise to the NADPH. Then NADPH, you see, when you have reactive oxygen species, what happens? See, what are they called? Glutathione. Glutathione reduces those reactive oxygen species, and then it forms, like, two of the glutathiones come together and they form a sulfide bond, a disulfide bond. So then now this NADPH is what breaks down that disulfide bond back to the glutathiones. Then it can keep reducing those reactive oxygen species. But then if now that is not there, so then the cells will just end up dying or something like that. Okay, guys, for these pathways, since most of the steps, I don't think there's a lot you can explain about the steps or such things. So maybe if, yeah, we can do some of the questions about the pathways today. Then we'll do, no, after we have done the other parts, which we'll be doing, what is it called? We'll be doing specialized tissues. Then after that, we can do maybe a few questions on the pathways. But yeah, so like we're saying, the pentose phosphate pathway can, depending on the body needs and demands, then it will be driven towards giving more of one of its products. So for example, like we said, if it's rapidly dividing cells, then we need more of the ribose 5-phosphate, as opposed to giving more glyceraldehyde, and maybe there's even no need for energy currently. So then it will be driven towards producing more of the ribose 5-phosphate. If maybe the cell is producing a lot of reactive oxygen species or such things, then you need more of the DH to cater for all that. Then you'd produce more of that. If the body needs both of them, then it just has to find a way to balance where it will give 2 NADPH, 2 ATP molecules, and of course, we'll still have the ribose being produced. Then, yeah, glutathione and how it's related to NADPH.
Seraphine, could
Yes.
you scroll a bit up where there was something about the ratio of NADH? I saw it briefly on the notes.
The ratio of what?
I think NADH to NADPH.
Oh, the
There,
ratio of
there.
NADPH.
Yeah.
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 So now for when we are trying to regulate the pentose phosphate pathway, see the pathway ends up producing more of the NADPH.
Yeah.
So if there is more of it, then it inhibits, like it stalls the pathway. If there is more NADPH and less NADPH, that ratio becomes big. Like so you're dividing a big number of a small number, it becomes big just by the fact that NADPH is a lot. Then that would inhibit the whole pathway thingy.
Okay. Sawa, tuko pamoja.
Yeah, and if the ratio is low, then it proceeds faster, like it's stimulated. Yeah, so glutathione is basically a reductant, and it can also be used to conjugate drugs. Yeah, but here we will mainly focus on it being a reductant and a cofactor in the enzymatic reaction, helping with the rearrangement of disulfide bonds, though that not so much here for but the disulfide bonds is what brings about a lot of those issues. So let me show you. Yeah, now this is what I was saying. So when you have those reactive oxygen species and they interact with glutathione, it ends up forming these disulfide bonds. And now you would need glutathione reductase to break these bonds. But glutathione reductase needs a cofactor to function properly. So if you don't have any NADPH, because maybe let's say your pentose phosphate pathway has an issue, then you cannot activate your glutathione reductase, then you can't break this bond here. And if you can't, then you won't be having enough amount of glutathione to now keep dealing with your reactive oxygen thingies. So then the cells keep dying and such kind of things. Yeah. What else? So we have seen the disulfide bonds when two of those glutathione things bind, but also it can happen with proteins. And if it happens, the disulfide bonds also happen in proteins and they are not broken, then they end up getting cross-linked, leading to the formation of Hen's body. And there we go with cell lysis. So that's basically about it for the two pathways that we were going to do today. So now we'll do, what is it called? The specialized tissues, then we'll do maybe a few questions on the metabolic pathways.
Seraphine,
Just give me a minute, kwanza.
Oh, sawa. That shout out to Fatma Shafi in the building. Na Kibunja.
Gaius, you were saying?
Could you like I want to just because we've talked about gluconeogenesis. I wanted just to highlight something, or because I know when we are covering it in pharmacology, it will be I want it to be a revision, just something small on gluconeogenesis that is related
Sure.
with what. Yeah, I don't know if you can just scroll up to where the PowerPoint, to where there's that picture of gluconeogenesis.
This one is okay.
Yes.
Yeah.
For broadcasting, oh, Seraphine,
Yes.
can you zoom out just kidogo? Just kidogo. Zoom out, not in, out, out.
Okay.
Yeah. So we can see the whole slide. Okay, a bit too much.
Yeah, let me try and balance it.
Oh, just weka 65. Weka 65. Yeah. There is better. That's okay.
Okay, there.
Now me I want just if you could zoom in just to the oxaloacetate and pyruvate, just there, and then you can return it to 65, because it's just there that I want to highlight currently.
What's the oxaloacetate and pyruvate?
Pyruvate, yeah, that step there.
Now someone here.
There, yeah.
The IT challenge.
Yeah, that's fine. So pyruvate to oxaloacetate, yeah. So I wanted to highlight metformin, because metformin is an anti-diabetic, which is actually the first line agent for diabetes, and one of its mechanisms is inhibition of gluconeogenesis, and so it inhibits gluconeogenesis by blocking this enzyme pyruvate carboxylase. It inhibits this enzyme here, pyruvate carboxylase. So once it has inhibited pyruvate carboxylase, our expectation is that pyruvate will build up. Then taking you back to chemistry, that we learned about equilibrium. It was that if one side, let's say, we know that pyruvate is usually converted to lactate and back, right? So if we've inhibited this pyruvate carboxylase, pyruvate is building up. So principle of equilibrium, we will start making more of lactate, more of lactate, more of lactate, until now, and we know when lactate accumulates in the body, we'll have this condition called lactic acidosis. Lactic acidosis. So that's the mechanism by which metformin causes lactic acidosis. Is just here. It inhibits this pyruvate carboxylase. Pyruvate builds up, which is then converted to lactic acid, causing lactic acidosis. So when we cover it again in pharmacology, I try to remember this. Yeah, thanks, Sarah.
Okay. But you're ready for us?
Yes, Seraphine.
So I can stop sharing?
Yeah, we're doing specialized tissue, right?
Yeah.
Okay.
Mmesikia metformin imeambiwa imepitwa na wakati, watu waanze kutumia semaglutide.
Semaglutide ni oral bado ama
Ni oral, wanasema ni DPP4. Hebu I'll share the link for that.
sawa.
Let me share it on the group.
Hi guys, can we start on the specialized tissue?
When you're ready.
When you're ready.
Sawa. So you already know these basics about how our body is structured at the chemical level, at the cellular level, organelle, then tissue. And then you have the organs, and you have the organ system, and then finally the organism, us. And we already know all the 12 types of so let's start with the biochemistry of the liver. Hopefully we already know the structure of the liver. We have the right lobe, we have the left lobe. Of course, the biliary system is there also. Yeah. And of course, it's surrounded by the Gleason's capsule, that's the tissue. The units, each specific unit, these ones, they are called lobules. Yeah. And then in between, in the middle, that's where the drainage happens. That's the central vein. Yeah. These are just basics. And of course, it receives blood from two major arteries. We have the hepatic artery, it supplies it with oxygenated blood. And you know, from all the digestive system, the portal vein supplies from the digestive system to the liver for the processes of the digestive molecules. Again, if you look into the central vein, what drains into this are these cells known as sinusoids. Yeah. Of course, we have other cells, just like all the other organs, which have specific cells. Even here, we have the hepatocytes, we have the stellate cells, we have the Kupffer cells, which are the different types of fighting cells, white blood cells in our body, in every different organs. Yeah. So hepatocytes we already know, which are our hepatic cell, liver sinusoids, Kupffer cells, which are part of the immune system of the liver cells. We have the hepatic natural killer cells, we have the stellate cells. These are in this piece of this. Yeah, this one, stellate cells. And And of course, this one again play a role in inflammation, being transformed into myofibroblast, secrete collagen. So again, part of immune system. Functions of the liver, we already know there are several functions of the liver, over 500 vital functions, and we have both non-metabolic and metabolic functions. So far, we have discussed most of the metabolic functions of the liver in metabolism, but the non-metabolic functions are like storage and release of nutrients, like they store glycogens that can release glucose into the blood, cholesterol, triglycerides. Of course, it also produces, it synthesizes and produces the bile acid, and that's why the biliary system is also there. It also produces certain blood plasma that functions to maintain the osmotic condition within the bloodstream, and also it does in the hemoglobin metabolism. We know about the xenobiotics, so clearing of blood of drugs and poisonous substances that we cover in xenobiotics. Also produces immune factors, regulates blood clotting, clears bilirubin. Metabolic functions, we've already done most of them, that is metabolism of carbohydrates. Maybe we've not covered lipid metabolism, but we can cover it here. Protein and nitrogen metabolism, cholesterol and vitamins. So those are the metabolic functions. Carbohydrate metabolism, we've already seen about glycolysis, gluconeogenesis. Yeah. So for the liver, it is very important because we already know at what range we should have our blood glucose to be, or else we'll either go hypo or hyperglycemia. Of course, diabetes kicks in. So we already know most of the pathways which are involved here, and the liver stores glycogen, which acts again as a buffer for glycemia. So again, as discussed above, when we're doing metabolism, it all depends on the body's requirements. So if you take a high glucose intake, then all that glucose will go into glycogen synthesis. But if the glycogen stores are overwhelmed, then most of the glucose will now be converted into fatty acids and stored as fat. So that's one thing that the liver does. And of course, we've already, Serafin and guys have already mentioned this, how there are different types of GLUT transporters. And in the liver, we are using, the hepatocytes are using GLUT2 transporters. And again, for any glucose to go under glycolysis, it must be converted into glucose 6-phosphate using the hexokinase 4, the glucokinase. So we've already talked about in enzymology how the enzymes have isoenzymes. So here we have the hexokinase 4 known as the glucokinase. And kinases are phosphorylating. They mostly add phosphates to the glucose. Once glucose is phosphorylated to glucose 6-phosphate, it cannot leave the hepatocyte. So it has to go under glycolysis. So that's what we've seen in enzymology, that glucokinase will have a higher km value for glucose than any other of its isoenzymes. So I've already talked about what is here in this slide, that any extra glucose which will be there is going to be converted into fatty acids and then to triglycerides. And some of the glucose 6-phosphate is also used in the pentose phosphate pathway. But all this will now depend on what the body's requirement will be. Yeah. So just today we did the pentose phosphate pathway. So you know that to enter pentose phosphate pathway, glucose 6-phosphate, the first product of glycolysis can just enter the pentose phosphate pathway and make for you your ribosis, and then further into nucleotide synthesis. Of course, generate any DPH. So again, when people are starved or they're fasting, the glycemia will decrease. And now what the liver will try to do is replace glucose in the blood, because you're not acquiring any glucose from outside. So that's where it will try to do glycogenolysis, which is breakdown of any glycogen which is stored. So that's one process which will happen if you are fasting. So liver hepatocytes have the enzyme glucose 6-phosphatase, which will cleave off the, so phosphatase will remove the, so one of the organs, or the only organ with this phosphatase, and will cleave off that phosphate so that glucose can go back into the blood. And this is necessary because brain is one organ which will only survive on glucose, and of course ketone bodies, but glucose is the most preferred. So the high km of glucokinase also ensures that hepatocytes do not use glucose as when there is low glucose concentration. In this case, mannose, galactose, and fructose are also converted to glucose 6-phosphate in liver hepatocytes and then used as a energy source. This is what we've been talking on Monday about the Cori cycle, what Hira was talking about also. So in prolonged fasting, if all your glycogen stores will become depleted, then the liver will start to produce glucose from other non-saccharide substrates like lactate, like glucogenic amino acids or glycerols. So what it will try to do is gluconeogenesis. So gluconeogenesis, as we've seen today, is synthesis of glucose from non-carbohydrate, non-carbohydrate or non-saccharide substrates. So that's where the Cori cycle comes in, whereby, and the glucose alanine cycle, whereby if there is no glucose in the body, then any glucose which is used by the muscle, which is converted into lactate, the lactate will go back into the liver again and be converted back to glucose. So that's where the Cori cycle comes in. And the same thing happens with the alanine. Any protein which is broken down into amino acid while during exercise by your muscles are also converted to alanine and then released in the blood. And then that same alanine, will be taken up by the liver and converted to glucose using the glucose alanine cycle. This alanine. So this graph also, it's a simple graph whereby we see that once you stop eating, any glucose that is in your blood will be utilized. First of all your ATP in the muscles will be utilized, then your glucose will be utilized in the blood, then glycogen stores will be depleted, and after glycogen stores, you will carry out gluconeogenesis, is try to make glucose from any other source. So you'll see that you'll start using most of your fat. And then once you see like what we see in Turkana, you'll start in the end, you'll start even using your protein like alanine, the way it will be converted to alanine, then to pyruvate, then to glucose. So that's where all these other diseases will come of starving also. It's called what? Kwashiorkor, I think. Yeah. So that will happen. And that is why you can see the difference between Usain Bolt and Kipchoge. He's running for longer hours, and that's why he carries out the whole respiration process. So he also uses his fat. So another important metabolism in the liver is the protein metabolism. So body cannot store amino acids, meaning that they must be broken down when in excess. And that's why most of the amino acids, you see them as urea. Yeah. So it will amine group, again, amine group cannot be amine group cannot just be released from the body just like that because it is toxic. That's why it has to be converted into urea again. So liver is responsible for the removal of the amine group, deamination, and its conversion into harmless product. And that's why it converts it into urea, and which is excreted in the urine by kidneys. And that's why you will see that in most of the chronic kidney disease, or even in the acute kidney disease, you'll see their urea levels increased, their creatinine levels increased, because the kidney is not able to filter the urea out, and most of the urea remains in the body. And that's why what was that called, if someone could remind me? There are those white snow-like flakes on the body which appears. So that shows that there is buildup of urea in the body. So it starts to show up on your face, on your body, on the outer surfaces. Urea is like white flakes. Yeah, and again, another thing to add on the part of how carbohydrate metabolism is occurring. You see, you'll also see with diabetic patients, most of them, you'll see that they're losing their muscle mass. So again, even though there is a lot of glucose in the blood, but it's not getting into the cells. So even then, this carry out a lot of gluconeogenesis and start depleting their fat and even their muscle mass. So the liver can also synthesize non-essential amino acid from surplus amine groups, so transamination. So the liver can either deaminate or transaminate to make the other non-essential amino acids which we don't get from food. No, which we get from outer source and we don't make them. Fat metabolism. So again, liver also does fat metabolism. It converts most of the carbohydrates and proteins into fatty acids and triglycerides. Remember, we cannot store amino acids, but we can store fatty acids. That's the best molecule in which we can store in the body and it will not be toxic to the body. So any excess glucose or any excess proteins will soon be converted into fatty acids, and that can be used later on when you're starving. It is also responsible for synthesis of large quantities of phospholipids and cholesterol, and which is very important because phospholipids make most part of all our cells and our cell membrane, and cholesterol will play a part in making most of our hormones, and that structure will play a part in making vitamins also. So it can be stored by the liver or also stored in the fatty cells in the lipoproteins. And we already know about LDL, the low density lipoprotein, high density lipoprotein. We also have the very low density lipoprotein. And of course, we know that LDL is considered as the bad cholesterol and HDL is considered as the good cholesterol. There is also plasma protein synthesis. Some of these plasma proteins are albumins, globulins, fibrinogens. We already know how these play a role in our body. Albumin being one of the biggest protein in our body in the blood, regulate the osmotic blood pressure. That's why you should never find albumin in the urine. If you do, then there is some problem with the kidney. Globulins participate in the immune system. That's why we have the IgG, IgM, IgE, IgA. Fibrinogens play a process in the clotting process. We have other plasma proteins like alpha-1 antitrypsin. antitrypsin, which neutralizes digestive trypsin, anticoagulants, albumin being the major part. Liver also carries out vitamin metabolism, yeah, and we know, for example, it converts carotene into vitamin A. It also stores some of the vitamins like lipophilic vitamins like vitamin B12. It also converts or activates some of the vitamins like vitamin D, which is converted, and it's one of the MCQs that vitamin D is activated at two places. That is one in the liver and the other one in the kidneys. Liver also deals with the trace elements metabolism. We know about our hemoglobin. So liver can once it has metabolized the hemoglobin, you need the iron which is left. It can make it into ferritin and then transport it back into the bone marrow, make more hemoglobin. It also stores other elements like iron, copper, manganese. Other metabolism which the liver carries out is purine degradation. We've seen that, and whereby we start producing uric acid, and that's where allopurinol and all these drugs comes in. Yeah, we've seen the xanthine oxidase coming in. We target some of these enzymes to reduce uric acid. There's also creatinine and choline synthesis. We already know about creatine, which is phosphorylated to phosphocreatine for excess ATP storage. We have choline, which is a precursor of acetylcholine, and also heme catabolism. We'll see heme catabolism again in detail, like here. So Kupffer cells in the liver catch and destroy old and worn out red blood cells and pass their components onto hepatocytes. Then hepatocytes will metabolize the hemoglobin into components heme and globin. And all this will be recycled and they'll be reused. Yeah. You already know the structure of the hemoglobin. So the iron containing heme group cannot be recycled by the body, and it is converted into pigment bilirubin and added to bile and excreted out of the body. But the globulin part can be reused. So heme oxygenase, this process happens in the smooth endoplasmic reticulum, and that's where we have the enzyme heme oxygenase, which does this heme metabolism. It requires heme as a substrate, and any hemine is reduced to heme F3 plus to F2 prior to oxidation by heme oxygenase. The heme oxygenase mediated oxidation occurs on a specific carbon using the linear blah 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 blah. So all this in simple terms, yeah, is that these we'll have to add this glucose. This chain needs to be opened up. You see here, this one here, we can on this hydroxyl group, it add the glucuronic acid. The 2-glucuronic acid become soluble. Can you guys hear me? Patrick, you can hear me?
Yeah, but we can hear you.
Sorry, I just internet connection. So I hope you guys are understanding. What is examinable is, what is examinable in the heme metabolism is mostly like the disease which will come, like uh, what do we really add to bilirubin for it to be excreted out? That is glucuronic acid. What colors they are, because biliverdin is greenish, while bilirubin in the end becomes yellow type, and that's why we have the jaundice. So those are the most important part. Another important part which is examinable is that uh, how is iron transported? Yeah, so it can be stored. It's stored as ferritin, but it is transported as transferrin. Yeah, so these are the things which are examinable as MCQs. So for this biliverdin to be converted now to bilirubin, which is now yellow in color, yeah, it is done using the biliverdin reductase. So here the first process was oxidizing, but here now we're reducing it. This oxygen will add an hydroxyl group, and it is NADPH dependent, because we need to add an hydroxyl group. So now you see where we generated our NADPH from all that metabolic pathways. We now also use some of them over here, as much as we'll also use it to reduce glutathione. We'll also use it to reduce biliverdin also. So these reducing factors which we've been generating are very, very important. Bilirubin again is highly lipophilic, and therefore it is insoluble. So that's why if someone is not able to excrete bilirubin out, if there's cholestasis, if there's blockage in that biliary system, you'll see bilirubin. You'll see their eyes to be yellow, their nails to be yellow, the skin turning pale. So because it's highly lipophilic and it can cross the barriers and be so there'll be a higher volume of distribution because it's highly lipophilic, and that's why this bilirubin will now be made hydrophilic, yeah, so that it can be excreted in the urine. So most of this bilirubin will be transported using serum albumin. Step 3. So on the smooth endoplasmic reticulum, bilirubin will be now conjugated with glucuronic acid, xylose, or ribose. In short, what we need to do is make it more of hydrophilic. Yeah. So 2 of them can be added here to make it hydrophilic, and which can now be excreted. It makes it water soluble, and then it can be readily be excreted in bile. So the process of conjugation can be induced by drug phenobarbital, barbiturate sedatives. Yeah. So this process again can be induced by phenobarbital. So I don't know if guys can add something on that. Guys,
Yeah, what's up?
this part here, I don't know if you can add something to this here, that the process of conjugation, when we are adding the glucuronic acid onto the bilirubin, can be induced by the drug phenobarbital. Are we trying to say that someone taking phenobarbital can be prone to having more of bilirubin formation or more of breakdown of heme, or what?
Yeah, I think it's they are prone to, there's more conjugation. So ideally, it's a good thing because ideally more bilirubin is soluble, so it's lost.
Mm,
Yeah.
okay.
Yeah.
So moving on, the last thing will be now excreting the bilirubin into the bile. So remember, it has 2 glucuronic acid, which will now be actively transported against a concentration gradient into the bile duct. So again, all that glycolysis, all that TCA cycle, all against the concentration gradient. So we also have other types of bilirubin, which is urobilin and stacobilin. So once you have some of this conjugated bilirubin in the intestine, it can be deconjugated using the beta glucuronidase and glucuronate. It can be converted into urobilinogen, and then which can further be reduced to stacobilinogen, and then the stacobilinogen can be now be excreted in your feces. So most of the will be so most of this stacobilinogen or urobilinogen will be oxidized by the bacteria to stacobilin, and that will give your feces the brown color. If intestinal flora is decreased by prolonged administration of antibiotics, and that's why sometimes doctors ask you what is the color of your feces, and if that antibiotic has removed your most of your normal flora, you end up having feces which have a bit of green tinge, yeah, because of the this reoxidized in the gut to biliverdins. So there are some factors which can influence your bilirubin conjugation. That is, it can be inhibited by hormones such as thyroid hormone and ethinyl estradiol, so estrogen. So again, that this can be a cause of increased bilirubin in your blood level. So it's not a must that there is a blockage or there is jaundice, like if there's anything to do wrong with the liver, but it's just the hormone which is inhibiting the conjugation. When it is inhibiting the conjugation, it's hard to also excrete it out. Combination of these contraceptives again can do that. Combination of the progesterone and the estrogen steroids can enhance UGT conjugation. Antibiotics such as gentamicin, novomycin also inhibit UGT at serum level exceeding therapeutic levels. Chronic hepatitis also inhibits conjugation. So in these cases, you might have jaundice forming. So bilirubin is taken up by the hepatocytes in the sinusoidal surfaces, and the albumin bilirubin bond is broken and albumin remains in the plasma while bilirubin can be taken up. It does require several different types of anion transport, and remember, it is against the concentration gradient. So again, it requires energy to do so. So most of the bilirubin is bound to ligandin, which is a cytosolic protein, and it facilitates the transfer to the endoplasmic reticulum and the site where the bilirubin will be conjugated. And then that conjugated bilirubin will leave out of the liver by the bile canalicus by multi-drug resistant associated proteins, and then into the small intestine. So this is more on the histology of how the conjugated bilirubin will move. But now this is the most important part where we can be tested. Is that the normal plasma bilirubin levels is between 0.2 to 0.8 milligrams per dl, in which conjugated should be that much and unconjugated should be less than 0.2. So if we exceed the level 1 milligram per dl, then that is hyperbilirubinemia. And if we exceed 2 milligrams per dl, then we'll have the condition jaundice. So some of the causes which increases the unconjugated bilirubin, that is if there is increased production of bilirubin production, and we've seen that with some of the, if you're using both the combination of contraceptive, you'll have an increased conjugation. If you're using phenobarbital, you'll have increased conjugation. If there is reduced bilirubin uptake by the hepatic cells, again they'll be increased in the. If there is also disrupted intracellular conjugation, there'll be increased unconjugated bilirubin. Factors which will increase conjugated bilirubins are disrupted secretion of bilirubin if there is a blockage into the bile canaliculi. So after it has conjugated and there is a blockage, then there'll be increased level of conjugated bilirubin. And also if there is an intra or extrahepatic bile duct obstruction. So jaundice can be in 3 types, that is prehepatic, that is hemolytic jaundice. You can have hepatic jaundice or post-hepatic cholestatic jaundice, which is obstructive jaundice. So if you are looking at prehepatic jaundice, which is also called hemolytic jaundice, meaning there's a lot of breakdown of the red blood cell, and there are so many conditions in which you'll have more of breakdown of the hemoglobin, and you'll see that is hemolytic jaundice. If there is internal bleeding, there'll be a lot of breakdown of the red blood cells. There'll be, of course, hemolytic jaundice. You'll see in that. If you see with some of the kidney patients, again, you have a lot of toxic buildup. Those toxic will affect the red blood cells again. You'll have a lot of prehepatic jaundice. If we've conjugated the bilirubin, but there is a blockage in the biliary system, and it's not able to release the bile into the intestine or into the bile. So again, that will be obstructive jaundice. So hemolytic jaundice, as I said, will be if there's a lot of massive lysis of red blood cell. So if there's a lot of massive lysis of red blood cell, there'll be a lot of formation of bilirubin. So that is prehepatic jaundice. A lot of unconjugated bilirubin will be elevated because the liver will not really have time to conjugate all that, or it will even deplete most of its gluconic acid. Hepatocellular or intrahepatic jaundice, it's where now there's a dysfunction in the hepatic cells. The liver will lose the ability to conjugate bilirubin. So an example is like whenever you have like liver cirrhosis, compression of intracellular caniculi at site of bile formation can also block the bile flow. So conjugation of bilirubin does occur, but the excretion will be impaired in intrahepatic jaundice. You can also have mixed type of jaundice in this case also. Plasma level of AST and ALT are also elevated, and the patient also experiences nausea and anorexia, tiredness. Hepatitis and chronic alcoholism are the major causes of liver cirrhosis, which can lead to this hepatocellular jaundice. Obstructive jaundice again is simple, it's just obstruction in the hepatic duct. So mostly conjugated bilirubin will be high in obstructive jaundice. For example, if you have hepatic tumor or bile stone, all the conjugated bilirubin. So the liver is okay, liver has done its work, it has conjugated the bilirubin, but there is a blockage, so it's not able to pass the bilirubin. And since no pigment are entering the JIT, the feces become clay colored, pale, and urine will be dark also. So again, bilirubin will be used as a diagnosis for jaundice. You'll see abnormal bilirubin levels in blocked bile duct, cirrhosis, hepatitis, and also immature liver development in newborns. Usually we measure the total bilirubin, direct bilirubin, and indirect bilirubin in serum and are thus determined, but indirect bilirubin will be calculated. So for direct bilirubin, we mean that direct bilirubin is the same as conjugated bilirubin, which is conjugated with the glucuronic acid, and non-conjugated is the indirect bilirubin. So if there's an increase in the indirect bilirubin, which is not conjugated, it's typical in the hemolytic anemia or the newborn jaundice. Increase of the direct bilirubin is mostly seen with the if there's any obstruction. And total bilirubin is manifold increased in viral hepatitis. So this is a test by which we check the bilirubin levels. So we use this chemical, which is diazotized sulfonamic acid, which reacts with the conjugated bilirubin and forms a purple colored complex, which is known as azobilirubin. Yeah. So remember that whatever we measure here will give you the measurement of your direct bilirubin, which is the conjugated bilirubin. Yeah, it is usually taken at 560 nanometer. So you can react your bilirubin with this compound, set the UV spectrophotometer at 560 nanometer, and then check the absorbances. The intensity of the color produced is directly proportional to the amount of direct bilirubin which is there. When you add methanol, it accelerates the reaction of unconjugated bilirubin in the serum, and value of total bilirubin is obtained after 5 minutes. So unconjugated bilirubin will be equals to the total bilirubin which you've gotten after adding methanol minus the conjugated bilirubin. So we do not test for the unconjugated bilirubin. We have to calculate that. So hemolytic jaundice will have more of the indirect bilirubin, which is unconjugated. Already talked about this. I hope you guys know about how bile is secreted. Kinin release estimated by the presence of Secretin again is secreted due to acid in the duodenum, and it stimulates the biliary ducts to secrete bicarbonate and water. 95% of the bile acid delivered to the duodenum are absorbed back into the blood. Venous blood from the ileum goes straight to the portal vein and hence through the sinusoid 2 of the liver. Some of the functions of bile acid, it is important in the digestion and absorption of lipids. Bile also acts as surfactants, helping emulsifying fats. We have some bile salts. Bile also serves as a route of excretion of bilirubin. It's also alkaline in nature, so neutralizes the acid. Bile salts are also bactericides, so they can destroy most of the bacteria. They also facilitate absorption of fat-soluble vitamins because they can solubilize this and prevent cholesterol precipitation. So we've already talked about cholestasis, where the bile does not flow from the liver into the small intestine, and that can occur due to liver damage or obstruction or defects in the enzyme making the bile itself. So two liver specific enzymes are usually assessed, which is ALT and AST. After you've done this, you can also measure GGT or ALP and also the bilirubin levels. So these are the most important ones. So alanine aminotransferase is an enzyme localized predominantly in the cytoplasm of the hepatocytes. So any damage to the liver cells, the hepatocytes, you'll see ALT leaking into the plasma, and hence you can measure ALT for that. The AST is also localized in the hepatocyte mitochondria, and any increase into AST can indicate more severe damage because now you're damaging the mitochondria itself. So any increase in ALT and AST, you know that there is some damage occurring to the liver. You can also go ahead and measure the GGT, yeah, and especially GGT after you measured ALT and AST, and you see that there's also an increase in GGT. You'll see that this can be an alcoholic patient, and maybe there's liver cirrhosis because of alcohol consumption. But remember that GGT is also increased in cholestasis, so you'll have to differentiate that. But if there is increased activity of ALP, that can also be a sign of bile duct obstruction or cirrhosis. So it all depends on the clinical patient that you have. Another examinable is the metabolism of xenobiotics. For example, ethanol. Ethanol, we know it is metabolized in two ways. We have the alcohol dehydrogenase, and we also have the cytochrome P450. Yeah. So alcohol dehydrogenase is the one which converts the ethanol to ethanol, an aldehyde, which then goes into the mitochondria, and then it's converted into acetic acid. So again, this alcohol dehydrogenase again is the rate limiting step, and it uses NAD. So the ethanol, it's converting into an aldehyde. So it has to remove the hydrogen group and oxidize it. Then in the end, the main thing is to make it more soluble, form it into acetic acid. So we know aldehydes can be toxic, and that's why we have to convert it into acetic acid. So most of this acetic acid or acetate can also be converted. Acetate-CoA ligase can be used in ATP-dependent process. Whereby this acetyl-CoA can be degraded in the TCA cycle to give you carbon dioxide and water, or it can be used in the synthesis of fatty acids or cholesterol. So we've already seen in the TCA cycle that pyruvate goes in the as acetate, right? As 2-carbon. And the same thing is used, acetyl-CoA again is used in the synthesis of fatty acids. Alternative pathway is where now we use the cytochrome P450 in our endoplasmic reticulum to also oxidize this acetic acid, the ethanol, into acetic acid. So this is where guys who are drinking heavily, their what do you say, their cytochrome P450 is what is doing most of the work for them, because ADH, the alcohol dehydrogenase, must be tired. So that's where the microsomal enzymes come in, the cytochrome P450. But to use the cytochrome P450, especially the CYP2E1, since it has a higher Km for ethanol, there has to be a higher level of ethanol for it to oxidize this into acetic acid. But of course, it also generates reactive oxygen species. And once you increase most of these reactives in oxygen species, can damage your hepatocytes, and again glutathiones and all this NADPH and all this will come try to reduce your oxidative stress which you created. So when you consume a lot of alcohol, there'll be increase in cytosolic and mitochondrial NADH, NH+. Will also be acetyl-CoA increased. This ratio will be decreased and NAD+ will be decreased. So what this will do is that when you're consuming alcohol, and if you go back to your glycolysis, you see that we've decreased this increased NADH and this decreased NAD+. This will mess up with the glycolysis, because there'll be that dysregulation which we've been talking about, and that will lead to a lot of lactic acidosis. Yeah, because it will start favoring more of the generation of the lactic acids. It also alters and affects the TCA cycle. It will suppress gluconeogenesis, suppress fatty acid beta oxidation. You'll see that in fat metabolism. You can see here that glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and this is occurring in the glycolysis. And remember that we have increased the NADH and decreased NAD+. Yeah, we have increased this NADH and decreased NAD+. So remember, it all depends on what is happening and what is there. Guys talked about that equilibrium. So if there's an increased NADH, this reversible reaction will of course favor upward. So you'll have more of glyceraldehyde 3-phosphate forming, and you're not carrying out glycolysis. You're reducing glycolysis. Again, the same thing. You remember pyruvate is converted into lactic acid. If you have increased NADH again because of alcohol consumption, you'll end up producing more of lactic acid because of that equilibrium. The same thing with the TCA cycle. One of the process whereby we are using NAD plus to NADH. You'll see that if you have increased level of NADH, of course, will form more of the malate. Malate will form more and not the oxaloacetate. So again, you're reversing this process here again. The same thing with gluconeogenesis. Yeah, since you're producing more of the malate, again that will affect the process. More of NADH will produce malate. More of NADH will produce malate, and then again oxaloacetate will be reduced. So again, TCA cycle is affected. Methanol, we know that methanol is toxic, especially formaldehyde. Will be highly toxic, can also cause blindness. And one of the best treatment, if one of our friend takes by mistake changa with a lot of methanol, make sure to buy him ethanol, maybe Imperial Blue. Ethanol will compete with the methanol for the same enzyme and convert into ethanol instead of the methanol. Acetyl salicylic acid. Again, our aspirin is also metabolized here, and glycine are what is added to it. So in the liver, conjugation will take place. Carboxylic group will be conjugated. These carboxylic groups will be conjugated with glycine, thus producing salicylic acid, which conjugates with glucuronic acid. So you can also conjugate with, apart from glycine, you can also conjugate with the glucuronic acid and then excrete it out. Are you guys with me, guys?
Yeah.
These
Yeah,
things are
but
simple.
we are here, but
Yeah.
it's if you can also try and just mention at least the very important points so that we can at least move. We have till 10 and I can see it's a few minutes to 8. So at least the key points, then we can move.
Okay. So as Serafin is stating, with the specialized tissue, the liver, yeah, the most important part are the metabolic pathways, which Serafin and Gaius really discussed well. Another thing is the how the heme metabolism and how bilirubin is produced, and how that part of the jaundice. And you have to know if it is pre-hepatic, post-hepatic jaundice. Those are very, very important part to know, because those are the ones which are examinable, right? And also with the ethanol metabolism, that's also very important. And especially the most important part about if there is methanol toxicity, what you can do, especially formaldehyde is the most toxic. And another thing is that a lot of alcohol consumption will disturb most of your metabolic pathways, as discussed. So those are the most important part. Muscle cell, again, we know that muscles usually utilize ATP as the energy source. Yeah. The three biochemical systems for producing ATP in order are the either you use the creatine phosphate, and you've seen our liver produces creatine for us, using glycogen, yeah, which is anaerobic glycolysis, and using aerobic respiration. Yeah, these are the main ways of that we produce. So creatine phosphate is just simple. If you have the the AMP or the diphosphate, you just take a phosphate from the creatine phosphate and make your ATP. Anaerobic glycolysis, we already know that will produce 2 ATP out of that. And aerobic respiration, we've seen that will end up producing 34 ATPs in the end, net. So this is a reaction whereby the phosphocreatine will add to ADP using creatine kinase. So we're just taking the phosphate and adding it onto the ATP and give you ATP plus creatine, and this will be recycled. So you're using creatine kinase, and the opposite will be done by a phosphatase. The same thing, we've seen this anaerobic glycolysis, which will give us 2 ATPs. Aerobic glycolysis, we've seen, which will give us up to 34 ATPs. So let's talk about epoch. When muscle fiber, when they're doing a lot of exercise, it accumulates with lactic acid. We know it depletes our creatine phosphate, depletes our ATP, and also our oxygen stores. Now to restore this, the respiratory system and the heart rate will remain elevated so that we can take a lot of oxygen, and so the epoch is the amount of oxygen required, which will replenish the myoglobin with oxygen. Also, it needs to produce the ATP, which is needed for metabolism of the lactic acid in the liver, heart, and skeletal muscle. Also, to convert lactic acid into glycogen, which is the Cori cycle, and restore any ATP and creatine phosphate. So, if a muscle is stimulated to contract for a long period, it will get tired and you're not able to contract that muscle anymore, and that will be that means you are fatigued, and that will be because of accumulation of lactic acid, um, lack of available nutrients, and of course less release of calcium. So again, as we talked in the liver, also here we have the Cori cycle. If there's a lot of lactic acid being produced by the muscle exercising, again that can be replenished as glucose, and it can come back through the Cori cycle back to the muscle. So that lactate gluconeogenesis can take place and can be converted into glucose and brought back again. So again, types of this is the important part. We have different types of muscle fibers, which is the slow oxidative fibers, fast oxidative, glycolytic fibers, and fast glycolytic fibers. So for the slow oxidative fibers, which also known as red muscles, they're the least powerful muscles and appear dark because they have a lot of myoglobin, have many blood capillaries, and this is where you'll be doing a lot of aerobic cellular respiration. So maybe these slow oxidative fibers are the ones which are used by Kipchoge. They have low glycogen stores. So they fatigue less, and they're mostly used by marathons like Kipchoge and slow cycling. We have the fast oxidative, which contain substantial amount of myoglobin in many blood capillaries, okay, but not that much of myoglobin as the slow oxidative. They also do aerobic cellular respiration, but also they get some ATP from their anaerobic, while the slow ones mostly do aerobic. The fast glycolytic fibers, the white muscles, they mostly use, they are very powerful. They have low myoglobin, and they have few mitochondria and generate ATP by anaerobic glycolysis. So they fast glycolytic fibers, white muscles. Maybe you can find them in whoever must be using it a lot. Some bolt. So again, this ratio of fast glycolytic and slow oxidative fibers depends on individuals. If you've been doing sprinting, so you'll have more of the fast oxidizing. If you've been doing long marathons, you'll have the slow oxidizing. We already know about the cardiac muscles, which do a lot, which relies mostly on aerobic metabolism. That's why the main source of its energy is mostly the free fatty acids, while for the brain is the glucose. We know the smooth muscle also, which form mostly the cavities. Yeah, that's about the muscle tissue. Do you guys want to go into the adipose tissue?
Yeah, but we can go through it in just briefly, like how you've done the one for muscle.
So this there must so the adipocyte cell structure will have, just like other cells, it will have some receptors on the cell. It will have a few of mitochondria, the nucleus, some GLUT transporters because it needs glucose to be converted into fatty acids and triglycerides, but it has this triglyceride storage. The neutral lipids are stored in the adipocytes in the form of lipid droplets with a core of stereoesters and triglycerols surrounded by the monolayer of phospholipids. The surfaces are usually coated with perilipins, which are proteins which restrict access to lipid droplets and preventing lipid mobilization. You will see that in a simple diagram. So we know that in the small intestines, how most of the what do you call them, nutrients which are absorbed there. So any triglycerides which we have or the phospholipids, they'll be packed into chylomicron particles like this one, whereby you'll have the cholesterol. Phospholipid will, of course, they'll form that structure by having the hydrophilic outside, the lipophilic inside. Yeah, that's the chylomicron structure. They will move from the intestinal lymph and then into the blood, which eventually carries them into the adipose and muscle tissues. There, the triglycerides will be hydrolyzed into free fatty acids and monoglycerides for tissue uptake by the lipoprotein lipase. So lipoprotein lipase here is the one which will take the chylomicrons, which will be converted into free fatty acids, which will be taken into the adipose tissue. The most important part about adipose tissue is this part here. And this will also take us to our, what was it called? The TCA cycle, right? No, the electron transport chain. So we have two types of adipose tissue. That's the white adipose tissue and brown adipose tissue. Yeah. The white adipose tissue is what we have, what is most common under the skin, abdominal cavity, deep blood vessel. But the brown adipose tissue are mostly found in small mammals, hibernating mammals like polar bears, or newborn humans, because newborn neonates do not shiver to generate the energy, but they actually have this brown adipose tissue which generates um, uh, energy for them. But us adults usually have reduced number of brown adipose tissue. So you can see the differences here. White adipose tissue have low mitochondria, while the brown adipocytes have high mitochondria. Yeah. There'll be a modest blood supply to the white, but there's rich blood supply to the brown. The white usually is for storage of energy, but the brown is to generate heat. This is the most important part. We've seen fatty acid. Most of the fatty acid we know it goes through beta oxidation to release energy and TCA cycle. We know how this, as explained by Gaius, how these electrons were generated, these protons were generated, and once they move in, the ATP synthase generates ATP for you. But this will not be the case for brown adipose tissue. Yeah. Yeah, this is the important part here. Thermogenin or uncoupling proteins, UCP1. This is a unique protein in the inner membrane of the brown adipose tissue. The protein will provide a unique route for protons to return to the mitochondria. So instead of having this ATP synthase, you'll have another protein, uncoupled thermogenin, which will allow these protons to return to the mitochondrial matrix without passing through the ATP synthase. So it will bypass the ATP synthase. And this protein, once it bypasses the ATP synthase, these protons, which when they enter, they release heat for the polar bears and for the newborn babies. So babies usually carry out thermogenesis in order to produce uh, heat, and that will keep them from hypothermia. So adipose tissue is the storage for triglycerides. It also serves as the largest energy reservoir for the body. Excess calories are also stored here. Again, this again the same thing we've been talking over and over again about what will happen in the well-fed state, what will happen in the fasting state. So of course, in the well-fed state, you'll carry out more of lipogenesis, you'll store more of triglycerides into fatty acids. And all this will be carried out by lipoprotein lipase, which will just present at the adipose cells just outside there. It will break these triglycerides into triglycerides, it has glycerol and 3 fatty acids. So it will just release the fatty acids and then take in the fatty acids and store them. So insulin is another hormone. Remember, hormones will play a very big role here. Insulin will always tend glucose to enter into the liver. It will also tend more of the glucose to go and be converted into fatty acids. So adipose tissue will convert the glucose via acetyl-CoA or malonyl-CoA to fatty acids. Yeah. And that fatty acids again will be esterified using glycerol to be converted and stored as fat droplets. So insulin will always increase the activity of glycerol phosphate acylate transferase. That's the de novo pathway, and that will convert glycerol 3-phosphate long chain acetyl-CoA to lysophosphatidic acid, and which is an intermediate of triglycerides. Insulin will inhibit the other enzyme, which is the hormone sensitive lipase, so that we do more of lipogenesis than lipolysis. While glucagon will do the opposite. Glucagon will increase lipolysis. So in fasting state, again, you're fasting, you need to release all your fat storage, and so that you can carry out gluconeogenesis. Yeah, fatty acids will be any released fatty acid will be carried by the albumin. So hormones like epinephrine will accelerate the release of fatty acid from adipocytes by stimulating the perilipid phosphorylation. Yes. So in case that you're fasting, or in case that you want to carry out gluconeogenesis, your hormone like glucagon will come, tell the adipocytes that we need fatty acids so that we can do gluconeogenesis, right? So these fatty acids will come. We know these processes like the G protein, the adenyl cyclase, this intracellular process which will occur. This is hormone sensitive lipase, which is because of glucagon, it will be activated. It will come, and a series of process will occur, which will release your triglycerides into into free fatty acids, monoglycerols, which can be released into the blood, and serum albumin will now transport it and take it maybe to the liver, take it maybe to the heart, where because heart uses free fatty acids. That's what I wanted to show you in general, not go into the detail of how each and every of these will function. Yeah. So most of these produce glycerol. So remember, the free fatty acid is what we need, but the glycerol will be used for glucose production in liver for gluconeogenesis. It can also be used for pyruvate production by glycolysis. So this is the moral of the story, that if you are well fed, you'll carry out more lipogenesis, you will decrease lipolysis. Insulin will inhibit the lipase enzyme, which is very sensitive to glucagon, and lipoprotein lipase will be active so that you can take in more free fatty acids and convert them into triglycerides. But during fasting, again, lipogenesis will be inhibited. You'll break down more of the lipids. The hormone sensitive lipase will be activated, and more of free fatty acids will increase in the blood. So again, the same thing, hormones playing a role. Functions of the adipose tissue. It contributes to thermal insulation. It protects the vital organs. Like you'll see the there is a fatty layer on top of your heart to protect it. Acts as a reservoir for energy. Also an endocrine gland because it produces the hormone like leptin, which controls hunger. Also there is non-shivering thermogenesis for the babies. Factors affecting the distribution, this distribution, yeah, with the genetic factors, gender and age, obesity and BMI. I hope all of us know how to calculate BMI, which is weight in kg over divided by the height in meters square. So obesity can be a genetic predisposition or because of the diet or having a sedentary lifestyle. But obesity can lead to some metabolic disorders like diabetes mellitus, dyslipidemia, and liver disease. Diabetes mellitus, we already know it's type 2 diabetes, whereby there'll be increased um, levels of sugar in your blood. And dyslipidemia, hyperlipidemia will also put you at risk of atherosclerosis, and this can lead to other diseases. Obesity is also leads to increased risk of non-alcoholic fatty liver disease. And if you do exercise, that can be reversed. Statio- Steatosis is accumulation of fat in the liver. It can also lead to cirrhosis. So the main thing about what I've seen in some of the MCQs is that they ask about this difference of the white adipocytes and the brown adipocytes, the thermogenin, the uncoupler proteins. That's what they ask about mostly in some of these questions. Yeah. Also, you must know, and it's simple, like what will happen to your adipocytes, what will they go under when you are in a well-fed state and when you are in the in the fasting stage. And again, you only have to remember two hormones, two enzymes, either the lipoprotein lipase, which will be active when you're in well-fed state because you want to convert, you want to break down in the blood and then take it up into the adipocytes and store them. And but if you are not well fed and you want to release some of those fatty acids, then the hormone sensitive lipase, and mostly hormones are playing a very big role. Insulin will play a role in well fed state, and glucagon will play a role in the fasting state. Also, epinephrine will play a role in fight and flight where you need some heart will need some energy. So again, that will activate your hormone sensitive lipase. You must know about some of the metabolic disorders, which is diabetes, dyslipidemia, and liver disease. And we know how diabetes, dyslipidemia, liver disease, what are the serious, how seriously it threatens the body functions. Should we go on to brain metabolism? Ladies,
We can take a 5-minute break.
okay, so Hi guys, we're back. Let's start on the brain metabolism. So, brain, most of the time it uses glucose-derived energy. Heart uses more of the fatty acids. Yeah. Why the glucose is required, you already know. Need energy to do many of the things in the brain. Yeah, you can read on that. We already know, we've gone through many times the cells. We have the microglial cells. Just like Kupffer cells, we have here microglial cells, which are the macrophage in the brain. Kupffer cells are in the liver. We have the oligodendrocytes with many dendrites. Yeah, we have the epidermal cells, the astrocytes. But what we are here to talk about is the metabolism. Yeah. So let's talk about metabolism in the brain. I want to take you guys to somewhere so that we can cover everything at once. Yeah. So in the brain, we are mostly using GLUT1 transporters, and you'll see we need to use the transporters because we have that blood-brain barrier, and most of the glucose will enter through the GLUT transporter into the astrocytes. Yeah, enter into the astrocytes whereby this glucose will go into glycolysis and convert into pyruvate. Yeah, that pyruvate using lactate dehydrogenase can be converted into lactate in the astrocytes, and that can again through this transporter be taken into the neurons, and that lactate can again be converted back to pyruvate. It can go to the TCA cycle and give you ATP. Same from the TCA cycles, we've seen that we can get glutamate, oxaloacetate. So many neurotransmitters can be also made from the TCA cycles. Yeah, because neurotransmitters are just made from some of these amino acids. Again, the same. This glutamate which we are making can also be recycled here. So we know that we are using GLUT transporters to transport, and usually we are using GLUT1. Neurons are predominantly oxidative, and so the neurons will mostly carry out the TCA cycle, the ETC cycle, electron transport chain, to give you the ATPs. But the astrocytes are more of glycolytic. Yeah, they will carry out more of glycolysis. And that's why we saw here that once glucose enters through the blood into the astrocytes will carry out more of glycolysis. And that glycolysis will give us pyruvate to lactate, and lactate will enter into the neurons, and then that lactate will be converted into pyruvate, and again it will carry out the TCA cycle. So neurons are more of will carry out the whole process of respiration, aerobic respiration, but astrocytes are more of carrying out the anaerobic respiration, just the glycolysis. So entry of pyruvate into the TCA cycle is upregulated by multi-enzyme pyruvate dehydrogenase. That PDH is activity is regulated by phosphorylation. Um, So neurons, they will predominantly process glucose through the pentose phosphate pathway to produce NADPH. Neurons will utilize substrates such as pyruvate, lactate, downstream of glycolysis to feed the TCA cycles because they want to do more of oxidative. But astrocytes, they will carry out more of glycolysis and produce more of the lactate and the pyruvate. So neurons will consume about 75 to 80% of the energy produced in the brain, whereas the rest is used by the glial-based processes. So since neurons do most of the function of the body, of the brain, they will take up more of the energy. So again, the glutamate produced glutamate can also be taken up by the astrocytes, converted into glutamine, yeah, and then recycled. So that will also be recycled using the excitatory amino acid transporters. So this again, increased transport of glutamate into the astrocyte will activate the sodium potassium ATPase channel and the conversion of glutamate into glutamine. And this process, of course, requires ATP to do so. So the consumption of ATP increases and its amount in astrocyte drops. And if in the astrocytes ATP drops, then that will again promote glycolysis, that it can produce more ATP into the astrocytes. So more lactate will be produced, and that will again go into the neurons, and that will be utilized there. So again, once it goes to the neurons, yeah, the neurons will use that pyruvate, that lactate to feed it into the TCA cycle, into the electron transport chain. And again, what is the main thing about neurons? It wants to make its neurotransmitters. These neurotransmitters, once made, yeah, they will also be recycled through the. You see, once you've released your neurotransmitter in the synaptic cleft, it will be reabsorbed by the astrocytes. It will be again recycled. For you to recycle that glutamate again, yeah, you need to use this ATPase channel, because this channel again, for it to take in glutamate, it takes in some sodium channels, it takes out potassium. So this sodium potassium, again, this channel will require ATP to do that, because it needs to balance out the sodium which comes in and the potassium which goes out. So that will stimulate production of that will stimulate the astrocytes to produce more ATP by glycolysis. I hope. And this glycolysis will produce pyruvate and lactate, which will go to the neurons. And these neurons will again utilize that in the TCA cycle. And that cycle continues. It will keep continuing. So that's the main thing to understand in the metabolism of brain, how it metabolizes. So let's talk about hyperglycemia and hyperglycemia. We've talked about what is happening in the normal physiologic condition. So if you have severe hypoglycemia, you don't have glucose, yeah. And if there is no glucose, yeah, again you will go to any reserve which is there, yeah. If the reserves are out, again you'll try to produce more glucose from gluconeogenesis to provide glucose to the brain. But again, if it's the opposite, which is hyperglycemia, that will increase the glucose concentration, and again that can worsen the outcome of stroke due to increased oxidative stress. Because and that's why it's not good to take soda or a lot of glucose at once, because most of this glucose will affect your cognition and memory due to conversion of excess glucose to sorbitol and fructose via the polyol pathway, which produces a lot of reactive oxygen species. Most of this conversion of glucose to sorbitol fructose will end up into producing a lot of reactive oxygen species, which will also deplete your glutathione, because the reduced glutathione will try to reduce your reactive oxygen species. Hypoxia, we again know that if there is lack of oxygen, you'll end up producing lactic acid, because you're trying to carry out more of anaerobic reaction. Imagine your astrocytes are already doing anaerobic, but you're producing a lot of lactate. But when you go to the neurons, there's no enough oxygen to carry out your TCA cycle or your electron transport chain. There is no oxygen to accept all those electrons and make water in the end. So there will be a lot of lactic acidosis in the brain also. And that can again lead to reduction in synthesis of acetylcholine. So again, some of these amino acids can be used to make some of the neurotransmitters which are amine in nature, like gamma aminobutyric acid, and also N-acetyl aspartate and cystathione. GABA, we know that it's one of the inhibitory neurotransmitter. Formation of glutamate from alpha ketoglutarate. Alpha ketoglutarate is coming from the TCA cycle, and in the neurons, you're carrying out your TCA cycle. So alpha ketoglutarate plus ammonia will give you the glutamate, and that is done by the glutamate dehydrogenase. The glutamine, which we saw, will get produced by glutamine synthetase. So this will be an ATP dependent, and this process will take place in the astrocytes. So astrocytes are producing ATP for this process also through glycolysis. Now we have the blood tissue. But the main thing about the brain tissue is to understand that astrocytes, from the blood you'll get from the blood. This is the main thing here, that from the blood through the GLUT transporters you'll get glucose. Your astrocytes will carry out glycolysis, and they will give you pyruvate and lactate to carry out to take it to the TCA cycle, electron transport chain, and that will produce a lot of remember I was telling you it will produce a lot of oxaloacetate, malate, all this alpha ketoglutarate, which will now be used as substance to produce now your glutamate, your other neurotransmitters. And then this recycling of the glutamate here, and remember glutamine to glutamate again is an ATP requiring process. So again, glycolysis will occur here. So this is a cycle. And that's all about your brain tissue. Should we go on to the blood tissue? Seraphine, should we continue to the blood tissue?
Guys, what do you think?
I think we should go through that like tomorrow. Like just a brief,
Okay,
an overview. Yeah.
tomorrow.
Yeah.
Yes, please.
Do you have some energy? We go through a few questions, or we also do that tomorrow?
Let's go through a few, Seraphim. At least those for metabolisms and we've done yesterday and today.
Okay, that's okay. You can just share. Then I'll send a few, the ones I saw in the book in the group.
Can you guys see my screen? Yes.
Yeah. We can see your screen, Shrina, among other things. Anyway, okay. Number 59, which of the following serum enzymes may be used in the diagnosis of myocardial infarction? So this one, Seraphine covered yesterday. It would be the creatine phosphokinase or creatine kinase, right? Because for the gamma glutamyl transpeptidase, that is GGT, that would be more of liver, so things like alcoholism. Then for the amylase would expect to see amylase if it has to do with pancreatic issues, right? Then for lipase, I don't know in what case lipase would be elevated. Maybe if someone was losing some fat, I don't. Seraphine, lipase. Anyway, we can move to the next regarding the effect of acetyl-CoA in lipid metabolism. Which one of the following is false? So acetyl-CoA lipid metabolism. Let me see if I can just send a picture of the lipid metabolism in a gist.
Yes, umesema 59 eh?
Spider-Man.
Sawa, Spider-Man.
Thank you. So getting acetyl-CoA is completely oxidized to so the cholesterol in the liver forms of lipogenesis to form amino acids. Lipogenesis to form so that statement is wrong. Acetyl-CoA may undergo lipogenesis to form amino acids. Lipogenesis is formation of lipids. So if it undergoes lipogenesis, we'd expect it to be forming those lipids, not even amino acids. So I think for that one is straightforward. Then hyperbilirubinemia may occur for all the following reasons, except which one? When bilirubin levels exceed that level. So assuming we don't know the limits of bilirubin, let's put that as a star. Due to production of more bilirubin than the normal liver can excrete, due to failure of liver to excrete bilirubin, due to obstruction of excretory ducts in the kidney. So hyperbilirubinemia may occur for all the following reasons, except obstruction of excretory ducts in the kidney. Is that, see, it's obstruction of the ducts in the liver, not in the kidney.
Yeah.
Yeah. So, these guys were just doing wordplay here. What about number 58, Shrina? I've seen something biochemical, like. Actually, we can continue going up. I will following statements below.
Gaius, just a question. The one with hyperbilirubinemia. See, we excrete bilirubin from the kidneys. So if the kidney has an issue, then wouldn't it cause hyperbilirubinemia?
But okay.
But see, the bulk of bilirubin is excreted in feces and not in urine.
No, it's
also excreted in urine.
But as urobilinogen, something like that, something.
Yeah, urobilin. Okay, I'm not sure, but it's just like I just thought about that.
Okay, my thoughts were, in that case, it would it be bilirubin that would rise in the bloodstream, or this urobilin or whatever? Because when bilirubin is reaching the kidney, it's not reaching as itself. But on the other hand, the reason I've thought it's also this, because I've seen it's like they're trying to confuse you. Usually hyperbilirubinemia would occur if there's obstruction of ducts in the liver. And higher, number 2, do we have ducts in the kidney or tubules? Yeah, I think here it's a word play. I think it's that one, ducts. We don't have ducts in the kidney.
Okay, that makes sense then.
Yeah, they were doing some serious wordplay. I'll go to 58. Oh, 50, yeah, 58, yeah. Which one of the given statements below regarding coenzymes is false? So coenzymes, remember coenzymes and cofactors. So most coenzymes have adenine ring joined to D-ribose and inorganic phosphate. Vitamin C forms part of the structure of many coenzymes. Chemical changes in the coenzyme counterbalance was taking place in the substrate. Many coenzymes may be regarded as derivatives of AMP. Now, for this, I think for this one you can use elimination. But or if you know, okay, the vitamin C is not true, but coenzyme, most coenzymes, they are without putting it like as critical as they have. I do not Shina, Seraphim. When you are covering coenzyme, I think you said most coenzymes, they are derived from nucleotides. So from that statement alone, A and D, we assume they are true because they, nucleotide moiety somewhere, adenine, AMP. Then, okay, from elimination, vitamin, we didn't mention vitamin C anywhere, but we mentioned that most coenzymes, they are derived from nucleotides. They are nucleotide derived. Then a polar amino acid. Now for this one, we agreed we just go and look at those structures, but it's the glutamic acid. At least for that one, when you see acid, or rather when you see glutamic acid, if you can remember, it's the one with the COH somewhere. But they are classified. So just remind yourself of the classification of amino acid, of the various amino acids. The polar ones, non-polar, aromatic, and such. Then all of the following are examples of problems with the something of transporting. Or what? Are examples of proteins involved in transport of, in the function of transport in the body, except, fibrin. Remember hemoglobin, heme, albumin, a lot of things transfer in iron, but fibrin is for clotting. Fibrin is not involved in transport, it's for clotting. This one we haven't covered, so there's apple. But I think we covered the last. I just go up a bit. A defect in dash enzyme may lead to gout, a clinical disorder of Nini. I think Bagwat mentioned this somewhere. So that hypoxanthine, guanine. Even there you can knowing the mechanism of action of allopurinol and how gout works, you can get that one for hypoxanthine. And all of the following statements regarding enzyme activity are true, except heavy metals like silver and magnesium are irreversible. Is that magnesium?
Mercury.
So it's mercury.
Oh, mercury. Oh, Hg, I was in Mg. Are irreversible non-competitive inhibitors. For competitive inhibitors at low concentration, those with lowest Ka values cause the greatest degree of inhibition. A reversible non-competitive inhibitor usually bears structural resemblance to the substrate. Let me pick this one.
Yeah, I also think it's D because if it's irreversible, you can't, when increasing the concentration of the substrate will not affect.
In a breakdown of molecules to release energy is called catabolism. Yeah. Anabolism is molecules coming together to form something. Then in glycolysis, glucose is broken down into, we said pyruvate and or lactic acid or lactate. Yeah, so that one is. Which one is an end product of carbohydrate digestion? Remember here we said it has to be a monosaccharide, that is glucose. Lactose is a disaccharide, it's still not the end product, so it's glucose. Function. Okay, functional properties of muscle tissue except I don't know if we've covered this, or we'll be doing it under physiology. Yeah, after that, these are just physiology questions. Yeah, I think it's still physiology. It's a bit familiar, but still physiology. Yeah, hepatitis. think. Yeah. Okay. Mitochondria, ribosomes are cell organelles. They are considered cell organelles. Kupffer cells, we just saw them right now. Hepatic Kupffer cells. Is it Kupffer? Albinism, lack of melanocytes. Yeah. Other ones, a series of amino acids are joined. Oh, polyuria, polydipsia, okay, we'll do that later. Series of amino acids are joined by peptide bonds to form a polypeptide, right? Well, is it phenylketonuria? Phenylketonuria is caused by the deficiency of is it usually phenyl what? Yeah, is that phenylalanine? Yeah. If I'm not wrong. Okay, then. Is it reductase or hydroxylase?
It's hydroxylase.
It's hydroxylase. Okay. Hydroxylase.
Yeah.
And the first step in the degradation of is it by the process of dash to yield alpha keto acid. Is the removal so is it transamination or deamination? Anyone? Seraphim, Shafi, Hiral, Patrick, Alakama is sleeping, Devayani, Nightwing.
I'm trying to figure out the reaction, because if I'm still trying to figure out that reaction, it can be either depending on the other parts of that reaction.
I've looked at a flow diagram. It is Nini. It's a transamination reaction.
Okay, so the transfer, okay.
Okay, or rather they've written transamination. But someone can confirm that.
It is transamination.
It's transamination. Okay, then. In humans, proteins are excreted as what? Urea. Yeah, you see this, yeah. Amino acids are precursors for the following compounds except proteins, nucleotide bases, nitrogenous compounds, lipids. Wait. Dietary amino acids are precursors for the following compounds except. Yeah, didn't see any amino acids can be converted to what? Okay, this one, help.
On number 71.
Yeah, is it 71? Yeah, 71.
Yeah, it should be lipids.
It should be lipids.
Yeah.
Okay. So does that mean if you eat just amino acids, you can't accumulate fat? That's okay. Done.
I'm not sure about that.
Then, okay, designation of monosaccharides into the D or L form is based on the configuration of which one? Glycerol, glycerin, glyceric acid, glysulfide. I could have guessed. And this guy knew everything. Not a disaccharide glucose. Which one is not a reducing agent? Which one the listed sugar is not? Ah, guy. Yo, we did this in high school. I think because of guys not participating, and we can just stop the discussion at that. But for the coming subjects, this lack of participation, I don't know. I don't know what you guys think about it. I feel like it's Iral, Shrina, Shafi, but just a few people, less than 10. And we said it was supposed to be more of a discussion. So,
Yeah, please,
and Seraphine was
everybody should should talk.
yeah,
Yeah, because it's a reason it's called
yeah.
a group discussion, not a few people talking and then that's it. There is no point then.
There's no point. There's no point if we be keeping up like this. So Seraphine wanted us to do 4 days a week from next week.
I agree with her. I feel this 3 days or less.
So you agree?
Yeah,
We do
Yeah,
Monday.
we can
especially
do
when
the
we
much
start with the
we can.
clinicals.
Like next week is microbiology, and that's like, oh my, so much. How will we finish
A
in 3
lot.
days?
Hey, by the way, because I'm the one doing micro, and I'd say at least 4 days would be good to that, because it's bacteria, viruses, fungi, and parasites. Hey, that's a lot.
Yeah, let's do 4 days. Those who are willing to join can join.
Yeah,
I
those who
don't
are
think we
busy,
can
there's
force
the recording.
anybody.